90% of drug candidates fail before final approval. That alone tells us something very important. The system is still spending a massive amount of money before it has enough confidence that it's pursuing the right biological path. 7 to 10 years, that has been the familiar rhythm of drug research. For a long time, long cycles, repeated experiments. Too much time and effort was spent just finding the right direction. What if biochemical research did not have to begin with the months of expensive trial and error? BinomLabs was created to solve this problem. BinomLabs is an AI-powered software platform that predicts biochemical in vitro experiments outcomes before broad wet lab testing begins. We help laboratories understand which experimental paths are most promising, which should be deprioritized, and where validation is more likely to succeed. We do not replace experiments, but we make experiments smart. This is especially important for infectious diseases research by making early biochemical research cheaper and more predictable. BinomLabs can help authorized teams accelerate systematic development of antiviral therapies, vaccines, antibodies, and diagnostics, including for the densely populated regions in Africa and Asia. Mostly important, BinomLabs has already successfully completed the proof of concept stage. In one completed proof of concept, a biochemical laboratory has already made 10 unnecessary attempts to reach the required experimental results. After receiving the BinomLabs prediction, the researchers needed only 2 additional experiments to achieve their scientific goals. This means our software moved from the theory to real laboratory impact. In validation work, BinomLabs also demonstrate up to 93% direct correlation with experimental data. Our advantages is not only prediction, but clear visual experimental relevant outputs that scientists can understand and use for their real decisions. Today, BinomLabs is on the pilot project stage. The software is fully ready for implementation and operational use, and we are opening pilot collaboration with research laboratories, biotech teams, and pharmaceutical partners. Our platform combines machine learning, structural biology, thermodynamic analysis, and direct comparison with experimental biochemical data. It can support protein-protein interaction analysis, antibody-antigen affinity prediction, protein stability assessment, mutation prioritization in viruses, and identification of biochemical path direction that are difficult to reveal through the standard experimental methods alone.